Well, good afternoon, everyone. I'm Jeff Harden from the Department of Integrated Biology, and I'm also a trainer in the Genetics PhD program. And it's my pleasure to introduce Yuyun Zhu. Yuyun is a senior graduate student in my lab, and what she's going to be talking to you about today is a project that is a collaboration with some former folks from my lab, and she's done some very important work to finish off that project, and it has to do with how cells change shape and what they have to do to their actin cytoskeleton as they do so. Yuyun also works on a completely different project from this, and you'll hopefully hear about that soon. And you've heard about that in previous colloquia in previous summers. I think that's all I want to say to introduce Yuyun, except to say that it's exciting to see how Yuyun's been able to take CRISPR-Cas9 genome editing technology and enhance the data that we already had related to this project. And I think you'll see how powerful that approach has been. Yuyun's title today is Tes and ZYX-1 Protect Junctional Actin Networks Under Tension from Self-Injury During Epidermal Morphogenesis in the elegans embryo. Yuyun.
Thanks, Jeff, for the nice introduction, and thank you everyone for coming today. And today I would like to share a story about how Tes1 and ZYX1 functioning during the epidermal morphogenesis to protect the actin networks in the C. elegans embryo. So first, I want to give a little bit background about this story. This story mainly focuses on one morphogenesis process, its embryonic elongation. So in the C. elegans embryo, we can see that the embryo will elongate from a coma stage to like fourfold of its original length within a short time period. Typically, it is like 3.5 hours. So as you can imagine, there will be dramatic morphological changes during this process. And more strikingly, there will be no cell dividing and cell movement, or also the embryo volume, it's almost the same during this process. So you can see that epidermis, which is the major driver for this elongation process, how much tension or pressure it has during this process. And here, our focus will be majority in the thin cell part, highlighted by purple here. We can see, I will introduce how Tes1 and ZYX1 regulated this elongation process or protect this elongation process. So here again, the purple part highlighted here is the thin cell, and the green line is indicating the actin part. We can see that there are some actomyosin within the thin cell, and also along the dorsal thin. Let's change it to. So you may also see this green line at the dorsal thin and ventral thin junction, and also a wellly parallelly organized CFB circumferential actin filament bundles along the epidermis of the embryo. So you can see this like green round structure. And since you may imagine the embryos are under like high tension to squeezing at one way, typically the dorsal ventral axis, and elongating and tweezing at the anterior posterior axis. So the cells must maintain its like junctional integrity during this process, and to make sure everything happens normally. And we are curious about what are the factors that can dedicate it to this process. And from a work done in Harding Labs about 10 years ago, it's a long time ago, we screened some, we did an enhancer screen experiment to identify some potential modulators that will be involved in this process. Here we are using the HUMP1 FE4 mutant, this sensitized background Okay, a little bit background about HUMP1, which is the homolog of alpha-catenin, and the FE4, this allele, will decrease the capability of HUMP1 to bind to the actin. So as you may see in the HUMP1 FE4 mutants, about 50% of the embryo will show these elongation defects, and there will be some cells pop out from the interior embryo, indicating the embryo cannot maintain a normal structure or integrity. And we are screening a bunch of genes to find out which one will enhance the phenotype, and specifically more than like about 80% of the lethality. And we find out something like Tes1, which is in the cytoskeleton related category from this enhancer screen result. So Tes1, we began to work on Tes1. And a little bit background about Tes1. This protein is highly conserved. When comparing the sequence between human Tes and C. elegans Tes1, we notice they are like highly conserved. And also the domain map analysis indicate that they are shared the similar PAT domain and also the link123 domain. And we believe here link123 domain will be a major component for the tension sensor. And you will hear that in the following stories. So did some experiments to verify the result and also see and check the phenotype. So from here, as you may see in wild type embryo, it just elongates normally. And interestingly, with Tes1 RNAi, the embryo looks quite normal. You can't see any like defects from this DIC scope. As for the HUMP1 FE4, about half of the embryo shown here will have defect during elongation, and treated together with Tes1 RNAi, we found that this defect will be enhanced to 100%, and also we observed some early defect compared to HUMP1 FE4, this mutant alone. So from here, indicating that Tes1 maybe functionally interact with HUMP1, and it also function during the early stage of elongation, since it promotes such a early defects during elongation. And since HUMP1 FE4 has some defects in actin binding, we are curious about, okay, except for the defects from the DIC movie, can our Tes1 RNAi lead to any other defects in terms of actin network? So here we are doing phalloidin staining using embryos from different backgrounds. As you may see in wild type, this really bright band is muscle. As for the actin structure, they are rarely like aligned into this. The structure you see here is the CFB, circumferential active filament bundles. As for in HUMP1 FE4 mutants, we observe some like clumping CFBs indicated here. And when treated with Tes1 RNAi, we recognize that the defect in HUMP1 FE4 is dramatically enhanced. So like indicated here, we saw way more clumped CFBs. And also you may notice such a like empty area indicating the embryo, no, the actin is detached from the junction. So, and the linkage of CFBs to the junction is critical when converting the force of contraction into coordinated elongation across the entire epidermis, and which is super important for elongation. And maybe this can explain why we saw such a dramatic 100% penetrase, considering the elongation defects. And then we are curious about how does if Tes1 bind directly with effecting or indirectly. So we did such a co-sedimentation experiment. Here from the quantification result show that compared to the negative control BSA, Tes1 shows such a significant higher binding level to affecting, and this level is similar to HUMP1, which is a well-characterized actin binding protein. So from here, we get the conclusion that Tes1 directly binds with affecting. And so we got to know Tes1 interact with HUMP1, and Tes1 directly binds to actin. And we are curious about if there is any defects in terms of the Tes1 loss of function itself. So here, another for phalloidin staining, and we look into the defects in a more detailed way. So from the wild type embryo, with the highlighted area, the blue rectangle indicates the CFP structure. It's well aligned and parallelly localized. It's really nice. And proximal, junctional proximal actin, we can see a great, a nice structure here. And we characterize such phenotype as type 1, which is the normal phenotype. As for the Tes1 OK103X allele, this allele is a French shift mutant, and it will has deletion in the link domain part, indicating it may have some defects in like tension sensing or linked to function together with actin. So here, for class 2 embryo, we can still see the normal CFBs, but with reduced junctional proximal actin. So compare these two panels, we just saw less level for the junctional proximal actin. That's class 2. As for the class 3, you can still see reduced junctional proximal actin and also the clumping CFB. As for the most severe part, class 4, you can see both clumping CFBs and also a severe detach of the actin from junction. So leaving some like empty space as for the actin network. And we did some quantification and showing, okay, so in Tes1 mutant, there's actually some defects in the actin networks, and indicating Tes1 can regulate actin networks during elongation. And these defects are consistent with Tes1 preventing damage to junctional actin networks due to the high pressure during or high tension during embryonic elongation. And then with all these phenotypes and defects set up, we are curious about the expression pattern and also the specific domain function of Tes1. So here, we use CRISPR-Cas9 to generate some knocking string and also specific deletion string to observe the localization pattern of Tes1. So the first line is the endogenesis knocking of Tes1 with amnion green. And the second line is we induce such a PAT domain deletion, leaving only link 1, 2, 3 domain here. As for the third one, we only have the PAT domain without link 1, 2, 3 domain. So by imaging these 3 strings, we got to see something interesting. So the first line is the amnion green Tes1 wild type string. Starting from the early elongation stage, we can see a like mired expression level of Tes1, and then the it will go specifically into the it's limited specifically in the syn cell, and also as the elongation happens, you can see some like recruitment of Tes1 to the dorsal syn and ventral syn junction. However, we saw something interesting in the specific domain deleted string. So, for example, in panel B, with delta PET domain with only link domain left, we saw a ectopic expression of Tes1 especially on such a Zyxin junction, which is absent in the wild type. And as for the delta link 123 domain, with only PAT domain left, we saw a dramatic expression pattern with Tes1 almost everywhere in the epidermis, including the expression in the syn cell and the syn cell structure. Also the CFB part, we can see some CFB similar structure here. And we also, since this observation is by eye, we also did some line scan analysis to check the expression pattern at the Zyxin junction and also the dorsal syn ventral junction. So within the deletion, the dorsal syn and ventral syn junction recruitment can still it's still there. However, the level may change a bit. The major difference is on the Zyxin junction and a overall increase the level of Tes1 expression. One possible explanation here is from some in vitro data showing that the link domain may have some like intermolecular inhibition function. So without the internal domain part, the Tes1 will be released from such inhibition mechanism. And also, things like something like in C, without the link domains like interaction, the PAT domain will simply bind to wherever it could. So we saw such a expression pattern. And besides the CRISPR-induced deletion or expression experiment, we also did the rescue experiment using some transgenic line by expressing specific truncated protein within the mutant strain. So here we are using Tes1 OK1036 allele with the FE4 heterozygous. We saw about 40% lethality in this strain, and this lethality phenotype can only be partially rescued by delta PAT construct, indicating the link domains of Tes1 are crucial for Tes1 function during morphogenesis. Since I say partially rescue, since the embryo still shows some defects regarding to other abnormalities. And later, we crossed the previously made Tes1 knocking strain with other reporter to specifically investigate on here the location, since we saw such a like dorsal syn and syn ventral specific junctional recruitment. Here we first crossed with the hump1, which is also the core member of, like I mentioned, which is the core member of cadherin catenin complex. We see such a nice overlap and colocalized instead of DLG1, another component of adherent junction. We didn't see a colocalization pattern, but overall here indicating Tes1 localized to apical junctions in the embryonic epidermis. And we are curious about if this recruitment can be disrupted by any factors. So here we introduce the HAMR1 RNAi and also the AGM1 RNAi here. So with HAMR1 RNAi, the embryos are they are lethal from like since super early stage. And we no longer see the junctional recruitment of Tes1 since it no longer elongated. The embryo never elongates. As for the AGM1 RNAi, there are some defects in terms of the junction structure. However, in this embryo, I showed the embryo still elongate, and we still see the recruitment of Tes1 to junctions. So we gave a preliminary summary from here is that Tes1 is only strongly recruited to junctions in cells that successfully elongate. And we did some further experiment to confirm the recruitment is tension dependent, not junction dependent, since when you delete hump one, that hammer one, that will affect junction severely. So from here, saying the AB shows the same sensitized background we used before, the hump 1 FE4, since only half of the embryo show elongation defects. So within those embryo has no defect, we can still see the recruitment of Tes1 to junction. But in those embryos showing elongation defects, we no longer see the recruitment of Tes1 to junction. And the quantification is only done in those embryos arrest at 1.5-fold stage, showing no difference, since the junctional and cytoplasmic signal here it's about 1, showing no difference comparing junctional signal and cytoplasmic signal. As for the lead 502 in this temperature sensitive allele, C is imaged under permissive temperature, so the embryo can successfully elongate it and has no trouble, and we can observe such a recruitment of Tes1 to the junction. However, when we shifted the temperature to a restrictive condition, the embryo can no longer elongate it, because lead 502 is important to provide the forces to help embryo elongate. So in panel D, the temperature no longer elongate. However, plus the junction, it's still like intact. However, we still don't observe, we still didn't see the recruitment of Tes1 to junction quantified here in the 1.25-fold stage. So from this data, we finally got to the conclusion that Tes1 will be recruited to junction in a tension-dependent manner. So when elongation happens, as I mentioned before, the tension, the junction is under like high tension, and Tes1 will localize to the junction. But if the embryo cannot elongate it with less tension trigger, the Tes1 will not relocalize to junctions. And okay, so since here we notice there are more than one LCR proteins, link domain containing proteins in C. elegans, and so we take a look at another LCR proteins, ZYX-1, which also show like highly conserved in sequence and domain structure. So here we did something similar, but here is using the transgenic line. ZYX-1 show a similar pattern compared to Tes1. It co-localized with hammer 1, the CCC complex, but not with the DLG1. This part is similar, and also the ZYX-1 GFP's junctional recruitment will be compromised by hump 1 RNA. And here this data indicates some similarity between ZYX-1 and Tes1. And we created a CRISPR knocking string and also the link domain deletion string to further investigate, okay, what's the localization or domain function for ZYX-1. So here, interestingly, we saw such a let's say complementary expression pattern between ZYX-1 and Tes1. If you still remember, the Tes1 is syn cell specific, and so the expression is only limited in the syn cell. However, for ZYX-1, we see the expression in non-syn structure, but the only possible overlap is at such dorsal syn and syn ventral junctions. And by removing link 1, 2, 3 domain, we no longer see such a junctional recruitment, and also Oh, one thing I forgot to mention here is ZYX-1 also show colocalized with the CFB structure, and in link123 domain deletion mutant, the ZYX-1 no longer show the colocalized with CFB. And also, like what we did in Tes 1, if we treated ZYX-1 knocking strain with lead 502, this will limit the tension. This will remove the tension, so the embryo can no longer elongate. And we noticed that the junctional recruitment level of ZYX-1 it's decreased due to such a defect in elongation. And interestingly, since Tes 1 and ZYX-1, they are both LCR protein, and they may function similarly in protecting junctions from high tension under high tension environment. Here, when we crossed ZYX-1 mutant in Tes 1 background or Tes 1 mutant in ZYX-1 background, we didn't see any change in the expression pattern, and also the co-localization data showing they are kind of not co-localized with each other. So we are guessing from here, Tes 1 and ZYX-1, they are act independently during elongation. They may function parallelly in protecting the actin network. And so those conclusions come from the in vivo data done performed in C. elegans embryo. And here we are introducing, we established the collaboration with an in vitro lab, and they are doing some experiment for us to examine such tension related function of Tes 1 and ZYX-1 for us in cultured cell. So just want to give a little bit background of the experiment. So in this fibroblast cell, the focal adhesion and the stress fiber, these two parts serves as a primary conduit for force transmission And when damage happens and the stress fiber has the locally in laser-induced damage makes the stress fiber locally weaker, and we may observe some factors be recruited to this stress fiber strand site and see how strong the recruitment will be, and that indicates the potential function of these link proteins. And here, they are transfected the cell with either C. elegans ZYX-1 or C. elegans Tes 1. At the same time, they have the mouse Zyxin as a positive control. So T0 means when damage happened. So when damage happened, we see such a significant recruitment of ZYX-1 to the damaged site, try to help the actin network and to seal the break. And when quantifying the protein level at the stress fiber strand site, we saw a similar level comparing the mouse Zyxin and the C. elegans ZYX-1. And we got the similar conclusion for C. elegans Tes 1 here. So when damage happened, both mouse Zyxin and C. elegans Tes 1 will be recruited to the same site, even though you may see a difference in the recruitment level. However, there is no significant difference between these two groups. So from here, we further validate our conclusion that both Tes 1 and ZYX-1 respond to tension and localize to stress fiber strand site by this in vitro system. So in summary, here we want to show that the LCR proteins, specifically Tes 1 and ZYX-1 in this context, they will be recruited to the junction when the junctions are under will be recruited to junctions under tension and protect junctional actin networks from self-injury during elongation. So since during elongation, the junction indicated here will be pulled up in a high tension. So you need some factors to jump in to help maintain the junctional integrity. So with that, I would like to thank the lab members in Harding Lab, and also forget to add Allison's name here, sorry, and also my committee member. And yeah, thank you for the time, and I'm happy to answer any questions.
One question.
Yeah, sure.
So in your deletion run in Tes 1, looks like one of those, I don't remember which one, but looks like that it's localized all over the cell.
Yes.
Compared with the white.
Mm-hmm.
So is that actually finding the actin, but it's not selectively choosing the actin which has a express or not?
Yeah, so this,
Do you understand?
yeah. So you are asking why with the deletion, it shows such a more colocalization with actin throughout the embryo.
I don't know if it's colocalized with actin, but my guess is that they are binding. So those the green
Mm-hmm.
probably binding with actin, but maybe not using the ability to select the different type of.
Yes, I guess so. I have the string, but haven't done the experiment to confirm if they are actually colocalized with actin or not. But yeah, and here, since we are thinking without the interdomain interaction, so there will be less limitation on overall binding choice. So the remaining part of the deleted protein will bind to whatever the feature induced. So finally, you see such a expression pattern.
So that the domain is kind of recognizing different type of activity.
I think they have the capability to bind to actin. Yeah. And I feel like I used too many time here. Any other questions?
Well, looks like you.
Hi there. My name is Peter Lewis. I'm an associate professor in the biomolecular chemistry department, and I'm here to introduce Andrew Rashoff. My lab is interested in gene management, genome management. Our research program seeks to understand the biochemical mechanisms that are involved in the establishment and maintenance of heterochromatin. And our experimental approaches, span the spectrum from highly purified biochemical assays to proteomic and genomic analyses and genetic screens. So today I'm going to talk about or introduce Andrew, who came here in 2019 after completing his Bachelor's of Science at UC Santa Barbara. After working in toxoplasma and Arabidopsis, he decided to move to a human and mammalian system. And he's been working on defining the chemical mechanisms of regulation of polycomb repressive complex 2, PRC2, which is involved in heterochromatin formation. So he's got some really exciting data, and I'll let him tell you more.
All right. Thank you, Peter, for that introduction, and thank you, everybody, for making the time to be here. I really appreciate it. So, like Peter mentioned, today I'll be talking about the mechanisms of inhibition of PRC2 activity within pediatric gliomas. So, in the broadest possible terms, the Lewis Lab is interested in the mechanisms by which cells with the exact same genetic background can differentiate into a multitude of different distinct cellular identities. A really important part of this complicated process is what has been broadly defined as epigenetic mechanisms. Specifically, what we're interested in is the chemical modifications of histone tails. So in order to package all of that DNA that's inside one single cell into the nucleus, an enormous amount of compaction needs to take place. And in order to do this, the DNA is wrapped around histone octamers to form the functional subunit of chromatin called nucleosomes. And so this DNA is wrapped around the histone octamer, but it doesn't only compact the DNA, it also serves as a robust hub of information used to regulate gene expression through the chemical modifications of the tails of each of these different histones. And a beautiful example of this taking place can be seen here in this in situ hybridization, looking at the expression of the HOXC8 mRNA within the developing embryo. So in the dark parts here indicating expression of the HOXC8 mRNA, and the lighter parts on the right side indicating a lack of expression of this mRNA. And so how this tightly controlled regulation of expression is accomplished is through the antagonistic role of two different groups of proteins. With the polycomb group proteins methylating histone lysine 27 on histone H3 to repress target genes, and the trithorax group proteins methylating lysine 4 histone H3 to turn on target genes. And so for my talk today, I'll be mostly focusing on these polycomb group proteins and their role in silencing target genes. So the core catalytic complex of the PRC2 complex is made up of 3 subunits. You have EZH2, EED, and SUZ12, with the EZH2 being the catalytic subunit of PRC2. And so an interesting aspect of PRC2 biology is that PRC2 can actually bind to its own product, H3K27 trimethylation, leading to allosteric activation of the catalytic EZH2 subunit. This leads to an increase in activity onto the neighboring nucleosomes, leading to the spreading of H3K27 trimethylation across broader regions of the genome. This repressive H3K27 trimethylation mark can then recruit other proteins, namely PRC1, to compact chromatin at these target sites, leading to gene silencing of target genes. So just zooming in a little bit on how this allosteric activation occurs, we have here in green the EED subunit, which is responsible for binding to the repressive H3K27 trimethylation. In yellow, we have the catalytic EZH2 subunit, which is responsible for catalyzing methylation on lysine 27. And then in the back there, you have SUZ12 in red, which serves as a scaffold for this entire complex. So upon binding of K27 trimethylation to the EED subunit, this information is transmitted to the catalytic EZH2 subunit through the stimulation response motif of EZH2. Then once this information is received, this leads to conformational changes within the catalytic domain of EZH2, leading to an increase in activity on this substrate lysine 27 there. And in vitro histone methyltransferase assays, this leads to about an 8-fold increase in the catalytic activity of PRC2. So as previously mentioned, PRC2 is incredibly important for development. However, dysregulation of PRC2's ability to silence target genes has also been implicated in a variety of diseases, and especially in many different kinds of cancers. Of particular interest in our lab is this recurrent mutation of the substrate lysine 27 on histone H3 into a methionine that's frequently found in pediatric brain tumors. So this H3K27M mutation is found in 84% of these diffuse midline gliomas and in 4% of these posterior fossa group A ependymomas. This H3K27M mutation is the earliest detectable mutation found within these tumors. And looking at these tumors using immunohistochemical staining, what you'll see is that within tumors that are positive for this H3K27M mutation, you see a global loss of H3K27 trimethylation within these tissues, as compared to a wild type tumor that has plenty of H3K27 trimethylation. And the same can be seen within these posterior fossa group A ependymomas as well, with less H3K27 trimethylation as compared to a related brain tumor. And so during Peter's postdoctoral work, he found that the only mutations of this lysine 27 that leads to a global decrease in K27 trimethylation within these cells is when you mutate this lysine from lysine into either an isoleucine or a methionine. And what these two residues have in common is that they have long unbranched aliphatic side chains. And so through very good work during his postdoctoral years, Peter found that these long unbranched aliphatic residues can form stable interactions with the aromatic residues of that catalytic EZH2 set domain, the catalytic domain of EZH2, and thus turning this substrate lysine into a potent competitive inhibitor of PRC2 activity through these stable hydrophobic interactions. And so upon discovery of this oncohistone mutation, this led to a greater appreciation for the role that histone post-translational modifications play in tumors, as well as a whole subfield of oncohistone mutations, with this lysine 36 on histone H3 being mutated into a methionine, being found in a vast majority of these chondroblastomas, as well as this glycine 34 being mutated into anything else, causing a giant cell tumors of the bone. Now, you'll remember that there was a small minority of these PFA ependymomas that also had K27M mutations. However, the vast majority of them do not. However, when you look at these PFA ependymomas, they universally have greatly decreased global levels of H3K27 trimethylation. They also show a incredible similarity in DNA methylation as well to these K27M positive gliomas as compared to other brain tumors that have been found in the brain. Another interesting piece of data is that these PFA ependymomas also express high levels of a novel protein that has been recently named EZIP. So EZIP expression within these PFA ependymomas is associated with an incredibly poor prognosis. And this EZIP expression is normally restricted to spermatogonia and is only found within placental mammals. Importantly, expression of EZIP is mutually exclusive within these PFA ependymomas. So there are some PFA ependymomas that have the K27M mutation or the EZIP, and there are some diffuse midline gliomas that can have EZIP or K27M, but you will never find one that has both. So when you look at the protein sequence of this EZIP protein, there isn't much sequence conservation across all of these placental mammals that have EZIP encoded within their genome, except for a very small portion of 13 amino acids that is completely invariant within all of these animals. When you take a closer look at this invariant sequence, you'll find that it bears striking similarities to the K27M oncohistone, as well as bearing striking similarities to what had been previously characterized as the ideal PRC2 substrate as measured by peptide arrays using histone methyltransferase assays. With the important caveat being that lysine, that substrate lysine has been mutated into a methionine within the oncohistone as well as an EZIP, which led us to the hypothesis that perhaps this EZIP protein is acting in a similar mechanism as the K27M oncohistone in competitively inhibiting PRC2 activity within these gliomas. And so when I first joined the lab, we were very excited to test this hypothesis and test this model. But before I continue, just a quick slide on rigor and reproducibility. So as everybody knows, you have to have your proper biological replicates, technical replicates, using orthogonal methods, et cetera. But I did want to put a little bit of emphasis on unique aspects that we have to keep in mind because of our cell culture models. So for one thing, you have to regularly test for mycoplasma infection within your cells. Mycoplasma is awful. It changes gene expression. It changes metabolic conditions within the cell. So you have to make sure you don't have any mycoplasma to make sure that all of your results are reproducible. We also have to keep in mind the differences in the genetic background of our cells that we're working with. Oftentimes, there can be differences in the genetic background or like the sex of these cells that can cause differences in different gene expression readouts. In addition, it's always important when you're thinking about mechanism within these different tumors that you use multiple cell lines to validate your results, because just because you get a result in one, say, glioma cell line doesn't mean that a different one is going to have the exact same result. So with that, let's get into the data. Like I said, when I first started, we were really excited because when you express EZIP within cells, you see, again, a global decrease in K27 trimethylation, much the same way that you see when you express K27M oncohistones. In addition, we found that when we perform a flag IP on flag-tagged EZIP, you can pull out all of the PRC2 subunits, suggesting that perhaps they're directly interacting. And when you follow this up with mass spectrometry, you'll find that there's amongst the most highly enriched proteins that come down are PRC2 components such as SUSY12, EZH2, and EED. Again, suggesting that these proteins are directly interacting with one another. In addition, when we use our in vitro histone methyltransferase assays, we find that adding that K27M-like peptide of EZIP is sufficient to inhibit PRC2 activity within our methyltransferase assays. And in fact, EZIP KLP peptides are an even greater inhibitor of PRC2 activity than the previously characterized K27M oncohistone peptides. And so next, we sought to find out what are the necessary parts of EZIP that are required for inhibition of PRC2 activity. So I made a whole bunch of truncation mutants. There's a lot here, but I'll just bring your attention to the fact that if you add everything C-terminal to the K27M-like peptide, it's insufficient to reduce K27 tri-levels, or N-terminus to the KLP, you also still see plenty of K27 tri. However, if you just add the KLP plus a tiny bit C-terminal to that region, you see that it's sufficient to inhibit PRC2 activity within cells, reducing global levels of K27 tri. Then, when we looked at the H3K27 trimethylation profile within these cells using ChIP-seq, we find that compared to control cell lines or cells expressing mouse embryonic fibroblasts expressing a K27R control, you see a global reduction in the amounts of K27 trimethylation in cells expressing both EZIP and K27M. And in fact, their K27 tri profiles look remarkably similar. However, I'm sure you all can notice that within these cells expressing EZIP and K27M, there are narrow peaks of H3K27 trimethylation that still remain. And this is really interesting because these cells are expressing really potent competitive inhibitors of PRC2. So you would expect that the H3K27 trimethylation would be completely flat within these cells. However, and one piece of data that's really important for potential explanations for this is that the allosteric activation of PRC2 actually increases the potency of inhibition within our in vitro histone methyltransferase assays. So within our methyltransferase assays, if you add stimulatory K27 trimethylation peptides, you see that the potency of inhibition is actually increased by 8-fold as compared when measured by IC50 value, indicating that EZIP is a more potent inhibitor in the presence of stimulatory K27 trimethylation. So this led us to the creation of a model in which, in the absence of allosteric activation, the binding between EZIP and PRC2 is relatively inefficient. However, when PRC2 catalyzes H3K27 trimethylation and then becomes allosterically activated, this leads to an increase in the affinity of EZIP for PRC2, leading to the creation of a high affinity ternary complex between H3K27 trimethylation, PRC2, and EZIP. And so we next went about going to testing this model. And so when we looked at mouse embryonic fibroblasts expressing EZIP, we find that looking at the EZH2 chip profile, the H3K27 trimethylation profile, and the EZIP profile, there's a remarkable amount of overlap between all three of them. And also shown here in this heat map, looking at all of the peaks where EZIP is found, you see a remarkable amount of overlap between PRC2, H3K27 trimethylation, and EZIP, supporting the idea that this high affinity ternary complex is being created. In addition, to further test this model, we wanted to ask what happens when you get rid of all that H3K27 trimethylation within the cells. Can EZIP still be localized? So to do this, I treated cells with tazemetostat, a pharmacological inhibitor of PRC2 activity. And what I found was, so with tazemetostat, you see a complete loss of H3K27 trimethylation. In addition, you see a reduction in EZH2 localization onto chromatin. However, there is still PRC2 found localizing onto chromatin. However, in response to tazemetostat treatment, you find that there's a complete loss of EZIP localization onto chromatin. And this can be shown here in this heat map as well, looking at all EZIP peaks again. And you find that, again, there's a slight loss of EZH2 localization, however, a complete loss of EZIP localization upon loss of H3K27 trimethylation. I next sought to directly test this hypothesis in vitro using recombinant proteins. However, just to back a little bit before I get into that, I just wanted to again remind you of how this allosteric activation is occurring. So the stimulatory peptide, upon binding to PRC2, transmits this information to the stimulation response motif, which then causes ordering of the catalytic domain of EZH2. However, an important residue for this transmission of information can be found on this arginine 302 residue of EED. And in fact, when you mutate this arginine into a serine, you'll find that PRC2 becomes completely unresponsive to H3K27 trimethylation stimulation within our in vitro histone methyltransferase assays, which makes this a useful tool in testing our model. In addition, when you ectopically express this R302S mutation inside of EED knockout mouse embryonic fibroblasts, you see that it fails to rescue global levels of H3K27 trimethylation within these cells. Furthermore, when you look at the H3K27 trimethylation profile within cells expressing either wild type or R302S, you'll find that the R302S mutation that is unable to become allosterically activated retains these narrow peaks of H3K27 trimethylation in much the same way that K27M does. And in fact, when you look at where these residual peaks of H3K27 trimethylation are found, you see that there's a remarkable amount of overlap between K27M expressing cells and this EED R302S, further supporting our model of inhibition of the allosterically activated form of PRC2. In addition, using peptide pull downs of pulling on EZIP peptides, we can find, using EZIP peptides to pull on recombinant PRC2, you'll find that stimulation of PRC2 with H3K27 trimethylation leads to a dramatic increase in the amount of PRC2 that's pulled down with these EZIP peptides. And using this EED R302S mutation that can't become allosterically activation as an orthogonal method in these peptide pull downs, you find that the inability of PRC2 to become allosterically activated leads to a reduction in the amount of PRC2 that gets pulled down within our in vitro peptide pull down assays. In addition, I also sought to use electrophoretic mobility shift assays to look at the binding of PRC2 with K27M nucleosomes. And so in order to do this, just to briefly give you an idea of how I did this, I combined recombinant PRC2 with nucleosomes harboring either the K27M mutation or the K27R mutation as a control. These nucleosomes were labeled with different fluorophores so that I could combine all of these into one test tube and let PRC2 reach an equilibrium with both types of nucleosomes, thus letting PRC2 figure out which nucleosomes it prefers to bind to. I could then run these on native polyacrylamide gels in order to see the differences in the binding affinity between PRC2 and K27M and K27R under a variety of different conditions in a highly controlled manner. So what I found is that upon addition of K27 trimethylation and a cofactor called acetylmethionine, which is the methyl donor cofactor of PRC2, what you'll find is that there's a dramatic increase in the affinity of PRC2 to K27M nucleosomes in response to it, as compared to when you don't have these cofactors, as seen here in the proportion of nucleosomes that are bound. In contrast, addition of acetylmethionine or K27 trimethylation has very little effect on the ability of PRC2 to bind to the K27R nucleosome. And in fact, if you're comparing this K27M nucleosomes without cofactors, it looks remarkably similar to the profile, the band shift that you'll find with the K27R nucleosomes. I can then use these band shift assays using densitometry to figure out the proportion bound under these different conditions. I find that the addition of these cofactors greatly increases the affinity of K27 of PRC2 with the K27M oncohistone nucleosomes. And again, since this is such a highly controlled system, I can look at the individual contributions of these different cofactors. For example, with PRC2 with or without acetylmethionine, if you add SAM in these assays, it exclusively increases the affinity of PRC2 to K27M nucleosomes and not K27R nucleosomes. However, if you add, if you're adding plus or minus H3K27 tri, what you'll find is that there's an increase in both in the affinity of PRC2 with both H3K27M and H3K27R. However, it's a relatively subtle increase in affinity. So just to bring this back to model I proposed at the beginning. So in cells that aren't expressing EZIP, what normally happens is PRC2 is recruited onto chromatin at these sites of initial recruitment, where it catalyzes the initial H3K27 trimethylation. And then upon catalysis at those sites of initial recruitment, PRC2 becomes allosterically activated and can spread H3K27 trimethylation onto broader regions of the genome. However, with the addition of EZIP in these cells, PRC2 can still catalyze H3K27 trimethylation at these sites of initial recruitment. However, upon allosteric activation of PRC2, you'll find that it forms a high affinity ternary complex, leading to this stalling of PRC2 and H3K27 trimethylation at these sites of initial recruitment. So I know there's a lot of nuances and getting into the weeds here, but for the last couple minutes, I just wanted to bring us back to why all of this is important in the first place in terms of within these pediatric gliomas that we're talking about. And an important piece of data that was collected by us and by others, this was by Tyler Reich in our lab, these diffuse H3K27M positive diffuse midline gliomas are actually sensitive to inhibition of PRC2, paradoxically. So tazemetostat, which I mentioned earlier, causes a drastic reduction in the proliferation of these diffuse midline gliomas that already are expressing this potent competitive inhibitor of PRC2, as shown here in these growth curves, as well as these pictures. However, these diffuse midline gliomas are also dependent on the oncohistone for their continued proliferation. So previous work has shown that if you knock out this K27M oncohistone protein from these diffuse midline glioma cell lines, you'll see a drastic reduction in the proliferation of these cells. And this reduction in proliferation can be rescued with the addition of ectopically expressed H3K27M oncohistones, but not K27R. And so this led us to the conclusion that the loss of PRC2 activity, such as when you add a drug onto these cells, is not functionally equivalent to the expression of K27M or EZIP competitive inhibitors of PRC2. In that these diffuse midline gliomas, these cell lines rely on the residual PRC2 activity from these diffuse midline gliomas rely on the H3K27M and residual PRC2 activity in order for the continued proliferation. And one previously characterized example of this can be found at the CDKN2A locus that is responsible for the expression of the P16 tumor suppressor. And so in cells that aren't expressing EZIP or K27M, you'll find that there is no K27 trimethylation within these cells. However, within these tumors that are expressing K27M or EZIP, you'll find that there's residual K27 trimethylation at the CDKN2A locus, thus shutting off the expression of this P16 tumor suppressor. So making, which provides a potential for why, or part of the reason why this residual H3K27 trimethylation is important for the continued proliferation of these cells. And the remainder of my thesis work will be focusing on the mechanisms by which this residual PRC2 activity allows for the continued proliferation of these pediatric gliomas. And so with that, I would like to thank Peter for being a great mentor, my lab mates for being awesome people to work with every day, my committee members for all their support, and I'd be happy to take any questions. Yeah.
overexpressed in food?
It's overexpressed in PFA ependymomas and diffuse midline gliomas.
And is it also developmentally regulated?
So the only places that ESIP is normally expressed is within the spermatogonia of, but its functional significance, besides giving children cancer, is still not really well known. So when you knock out ESIP in mice, the females show a reduction in fecundity, but yeah, I know it's weird. We still don't really know what ESIP is actually doing, like why we have ESIP at all. So that is, there are some RNA-seq data sets that have very, very, very low levels of ESIP expression, but its functional relevance, we have no idea. If it's, you can find like tiny amounts of it in normal brain tissues, but very negligible compared to like spermatogonia. No, exactly. There's still a lot of questions to be answered in terms of what ESIP is actually doing outside of giving people cancer. Yeah, Chris.
So the glucose system, I'm curious in the HG827M, those are always heterozygous, I assume.
So yeah, good observation. So there are a lot of different copies of H3 found within a mammalian cell. So the total amount of K27M protein in terms of like the amount of H3 within the cell, it's around 1%. But it's such a potent inhibitor that it leads to the global loss of K27 trimethylation.
Are there ever any gain
So I don't know of any of like recurrent aneuploidies. There are recurrent mutations that you find, such as p53 deletion and increase in expression of PDGFRA. I don't know of any chromosomal aneuploidies that are, yeah, I'm trying to think.
If there are any structural variants associated with H3?
Wait, sorry, say it again.
If like in terms of changing the ratio, H3, H27M, H3, the wild type, do they ever have any?
Not as far as I know. Generally, it's just a one copy that's mutated at a very, the model is there's one copy mutated at a very specific developmental window that leads to.
It's legal earlier. You can't be, if you have a single copy of this, is there any population level polymerase disease variants, or is it all just?
I think it's all disease associated. Yeah, I can't imagine being able to develop properly. Polycom's too important for development of embryos and things like that. So
Then I kind of thought the explanation for P16. The idea is as an explanation of why these are proliferating with their current polycomb expression is because mostly it's due to the reduction of indivision or the reduction of expression of P16, which is
that's one aspect of it. I'm currently studying. There's more to it, certainly, but it's that basically there's this Goldilocks zone of K27 trimethylation, and you need that K27 trimethylation at residual K27 trimethylation at specific genes in order for continued proliferation. So if you have too much and you start spreading K27 trimethylation across the genome, then you're shutting off genes that are important for continued proliferation. However, if you get rid of all the K27 trimethylation within the genome, then you're allowing for expression of things like P16 that would stop the proliferation of these cells.
So have you guys been known, like you know the targets of de-repression that are, why is this, what is being expressed by this expression?
So lots of things, because you have a global reduction in K27 trimethylation. So there are a lot of things that are being de-repressed. There are some known, like platelet-derived growth factor alpha is known to cause the increase or overexpression of PDGFRA is found within many of these K27M gliomas, but there's certainly many genes that are being upregulated in response to K27M expression. Ben?
So lots of super great, clear biochemical work here. My question, I don't know if you've looked into this, but are there ideas on what allows EZIP expression to increase in some of these cells, like
DNA methylation. Yeah, so previous work is from other labs showed that if you use 5-AZA, which is an inhibitor of the DNA methyltransferase, then you can see increase in EZIP expression within a variety of different cell lines. Yeah, so if you remember, I won't go back, but there's a very specific DNA methylation profile within different gliomas, different types of brain tumors. And that's almost certainly playing some kind of role in what's allowing this vulnerability for EZIP expression to occur. Or I would hypothesize that, and I would good reason to believe that. Great. Anything else? Sorry, I'm a little over time, but thank you all for being here.
Thank you.
Oh, thank you.
I have a basic biology question.
Yes.
What breaks the autocatalysis or the positive feedback?
Yes.
Island latches on?
So other histone modifications. So specifically K36 methylation is associated with active transcription. So there's differing levels of like, there's monodi and trimethylation. And so like trimethylation is associated with active transcription.
And acetylation, how about, is that?
We mostly, it's a lot of recent work about like what role even is acetylation playing in gene expression. So it's hard to say exactly, because like there have been studies where like if you get rid of all K27 acetylation, nothing changes with gene expression.
Oh, no kidding. Wow. Okay.
So, but like I think it's we have lots of thoughts on that, but like for example, one of the binders of K27 acetylation can also bind other acetylation marks. So perhaps even though it binds K27 acetyl the best, perhaps this transcriptional activator that binds K27 acetyl can also bind other acetylation. So it's sort of like there's a acetylation redundancy, perhaps. But we think that changes in, because when you have K27M, you see increase in K27 acetyl. And we think that's a byproduct of PRC2 not being able to act rather than actually having any kind of functional relevance within these gliomas. Yeah.
Well, all right. Well, thank you.
Sorry. Yeah.
Appreciate it.
Thank you. I don't know if